First, I want to thank Bjorn and the rest of the student postdoc committee for organizing this session on open science. I'm honored to be giving a quick introduction to the topic for the Vision Science community that I'm proud to be affiliated with. Here's my vintage 2011 VSS t-shirt. I want to start with something that Newton basically wrote in a letter to a friend 400 or 500 years ago. If I have seen further than others, it is by standing upon the shoulders of giants. Now, besides just being humble here, what Newton was indicating was that science is cumulative. He was only able to make his advances by knowing about the results of others. And thus, sharing has always been an important part of science. We don't keep our discoveries secret. Rather, we like to actually tell everybody this amazing discovery we found, whether sometimes maybe it's for our own self-aggrandizement, but the more charitable aspect to it is that we are trying to advance knowledge together, and so we need to build on each other's results and thus share what we're doing. Robert Merton, the sociologist of science, almost 100 years ago now, recognized that scientists have a value that he called communalism, by which he meant that we do, rather than keep things secret, share things. And of course, because science is cumulative, sharing speeds science. Merton also suggested that scientists have the value of universalism, by which he meant that scientific findings, we don't evaluate their credibility or count them as science or not based on who does them. Rather, we are focused on the evidence, regardless of who's doing the work. Nowadays, we might call that equity, diversity, and inclusion, but you should recognize that in science, it goes back hundreds of years as a core value. So these are sort of a motley group of values here, but we can group them under the word openness. And so that's how I think about where the term open science or open scholarship came from. We've got certain values that align with what we're trying to do, and thus we have this umbrella term, open scholarship, that refer to the sharing practices for data, for our method sections, for our experiment code, analysis code, and just the work itself as with open access. But historically, we have not lived up to these as well as we might, and so we have not lived up to our values, because part of it is probably just for historical reasons that, with journals, when they started, it was all about only being able to communicate as much stuff as you could print with the printing press. And with limited pages, what we ended up with is this journal article format that describes the results as sort of a summary without providing every individual data from individual trials, in the case of vision science, or providing, a copy of the equipment, or in today's terms, a copy of the software that was used to run the experiment, or the exact step-by-step data analysis. We might just say an ANOVA or something in the method section. But now we shouldn't be so limited, and thus we can potentially advance science more quickly by sharing the details of these things so that people don't have to redo them for themselves when they're trying to build on something. So I think of open science practices as speeding science while furthering some core values that we have more broadly, such as this expectation that anyone should be able to contribute to science. We're not some kind of elite priesthood. And this does go way back. The Royal Society, when they created this logo for themselves, this coat of arms, on the bottom they emblazoned nullius in verba, meaning take nobody's word for it, which is, part of this whole universalism idea, that science is not about authorities handing down facts, but rather that we are what Richard Feynman and Robert Merton described as a community of organized skepticism, where our scientific results gain credibility because we have other skeptics looking at them. And part of that is reproducibility, which can include what I call empirical reproducibility here, where someone comes along and tries to replicate an experiment, and they may replicate it, but sometimes they won't. And this, really became conspicuous over the last 10 years when, for the first time in the history of science, people started trying to do systematic studies of what proportion of scientific results could be reproduced. And it led to some pretty disappointing findings. People call this the replication crisis, but is it really a crisis? Maybe judge for yourself. Let me show you some numbers. So I divide reproducibility into three portions. You can think of just whether the methods are reproducible, meaning is there enough detail provided in the method section, or hopefully additionally the actual software code is provided for various aspects of the experiment such that another researcher can fully reproduce it. There's also what I call computational reproducibility, which is if you take the data that someone collected and you look at the statistics that they described and the main results, the plots and so on in their paper, can you reproduce those by rerunning the analyses from their data? So that's computational reproducibility, or just computing the numbers again. And then empirical reproducibility is where you go out and you collect new data. When you do so, if you collect new data and you rerun those same analyses, will you get the same result? So this has now been studied across various fields, not with representative samples necessarily, because that's very hard to do. So these numbers are going to be just very indicative and rough. But when it was done in economics a couple of years ago, someone tried to look at just of those economics papers for which the data is publicly available. Often they are in some kind of public database and so on. They tried to rerun the analyses, but they were only able to get the same kind of results that were reported in the paper for 55% of the papers. Now there can be various reasons for that, but clearly one of them is going to be that the exact data analysis scripts were not shared, and so it's very difficult to be able to know the exact details of how these complicated regressions were run, or other models, statistical models, without that. And so that leads to some of the failures to computationally reproduce. Of course, some of them are instead due to errors that the original authors made. But lack of sharing is one cause. Now, there's not just the reproducibility project psychology that's the most famous case, where something like almost 97 psychology articles in leading journals were attempted to be reproduced, but also there's been many other many lab studies. And if you sort of roughly average the results from those together, you get this disappointing figure of less than 50% of replication attempts that have been done have led to the same finding, by which I mean they got the same statistically significant results as were reported in the original study. Now, this should really concern us. And, it's not just confined to just say like psychology journals. Colin Kammerer and others, they studied the social science findings that were published in Science and Nature, and they really avoided the statistical power problem that could be responsible for some of the failures by boosting the sample sizes way beyond the samples that were used in the original studies. But still, they didn't do that much better than 50%. And turning to method sections, that's particularly an issue in very complicated procedural sciences, like maybe wet lab clinical biology. You've got all these different machines in your lab, and you have to run them step by step, and the gels and the gas chromatographs and the centrifuges. When Tim Arrington and others at the Center for Open Science tried to replicate, I think it was 50 papers, they found they couldn't even get started, because for 0 of them were they able to develop a full step-by-step methods protocol for their lab collaborators to just run, to do. And you can imagine that with VSS being a really diverse field, where you've got some areas that just might take mTurk or some very simple online experiment, that's maybe a straightforward procedure. But others, for example, with neuroimaging or with complicated equipment related to ophthalmology and so on, the method section needs to be much more detailed, and it's easy to miss some steps because no one's none of the reviewers are actually trying to follow the steps step by step. So this can be a problem in the fields that vision science is involved in as well. And we know that there's going to be many reasons for these disappointing results that I think do deserve the term crisis, because they should motivate us to address them. There's well-known threats to reproducibility that many researchers, a high proportion across multiple fields, will admit to, such as publication bias, meaning that people tend to not report negative findings or findings that don't comport with their theoretical predictions, and thus some people keep rerunning experiments thinking, oh, well, that probably didn't work because such and such, but I know if I run it again, I'll get the result that I'm predicting or that I want. But that means that findings appear to be more robust than they are, and some of those published findings are going to be false positives. Similarly, for chasing statistical significance, we've got p-hacking, which is commonly admitted to, at least historically and actually very recently, by researchers across multiple fields. That's going to litter the field with false positives. And unfortunately, due to when we don't have sharing of data and code and stuff, it's hard to even evaluate whether this is happening. But we do know that these forces, these pressures on us as scholars to produce, publish. And so some of this is always going to be happening. So to get people to practice these things, behaviors that can lead to better evaluation, these badges were developed by the Center for Open Science. The way that I think about sharing one's data is that it increases credibility, because it's a message basically saying, here, you can check our data. We're confident in it. Yes, unfortunately, approximately only 50% of results, at least in those fields, have been studied, replicate. But so, we need to do what we can, and here I am showing my data so that you can at least evaluate computational reproducibility and thus have higher credence in what I'm reporting. Or I'll provide my software, post that to GitHub or the Center for Open Science website, meaning that you can try the experiment yourself and thus verify what I'm saying in the method sections matches what I did. Here's my R scripts or MATLAB scripts for doing my data analyses. And finally, pre-registration. This is going to have varying importance depending upon exactly what you do, but I think of it as importantly providing a signal that one did not p-hack. That is, I didn't just try lots of different data analyses until I found the one that was going to give me a p-value of less than 0.05. And I think that's important because even in relatively rigorous fields, so if you ask maybe 8 different researchers how you should analyze data, ask a question, you might get 8 different answers. But if you know that that person was planning this analysis from the beginning, that gives more strength to what the p-value implies than if you don't know whether they actually analyzed the data 8 or 10 different ways. Now, in some field like theoretical physics, where you've got very formal theories, maybe you can derive quantitative predictions such that you know exactly the way, and the reader knows exactly the way the data should be analyzed just by knowing the theory. But that's rarely the case, I would say, in vision science, neuroscience, psychology. So I'm going to use these symbols over here on the right because I want to show you some data that Steve Hauerhaus and Charlie Ludovici and I collected, by which I mean we looked at lots of different Journal of Vision articles over a few years. We looked at every article in these issues, and we checked whether Steve wrote code to scan the text of the articles looking for URLs, which helped us more rapidly scan the text to pick out where links were provided that might link to open data or the software that was used to run the experiment or the scripts that were used to analyze the data. And what we found is that these open practices have very low uptake so far, at least in Journal of Vision. So each of these X's an indication that one article in one issue did not do any of these practices, or each of these rectangles, I should say. So there are a few scattered articles that did pre-register or share open data, share their software, share their data analysis code, but it was few and far between. So whatever you think of how bad the reproducibility crisis might be in vision science, and personally I don't have much sense of that. We have such disparate methods, some of which have very high statistical power within participant studies where you're running for many hundreds of trials, others of which do not have high statistical power. So it's hard to make a summary judgment, but you should expect that the reader of these things, especially knowing about the reproducibility crisis elsewhere, will not have very high confidence that our results will replicate. And maybe they're right. Either way, even apart from whether these studies all replicate, we should be sharing our data, sharing our software, sharing our code, our analysis code to advance science more quickly. But we're a bit behind the vision science community relative to certain areas, for example, of psychology. Here, Katie Corker, who is on the Open Science Committee, is describing how the American Psychological Association, which publishes 29 journals, have signed on to these transparency and openness promotion guidelines, meaning that they've instituted a policy to either encourage or in some cases require data sharing, code sharing. And so they're busy building up the credibility of their field. And I'm afraid we're a bit behind on that. So I encourage you to have a look at that. And if you're associated with a journal, scrutinize these transparency and openness promotion kind of example policies for which there's 3 different levels of requiring each of these different things. And I hope we're going to see more and more of that adopted at vision-related journals. But part of doing that means that we need to adapt some of these practices for every specific subfield, which has been going on a lot in neuroscience with standardizing, let's say, EEG analysis and fMRI pipelines to make them more reproducible. Now, in classic psychophysics, we have this great situation where we're often able to collect a huge amount of data and within a person such that we can establish our finding within individual people and then think of each additional participant then as a replication. So we're actually replicating all the time in certain subfields of what you see at VSS. But the psychology journals that some of us publish in aren't thinking of that kind of design, and so their policies were not written with that in mind. And so I would encourage those of you who want to advance this to check out things like this Society for the Improvement of Psychological Science conference, where me and a bunch of other vision researchers at the last in-person meeting. And this year we're put together a hackathon, Sarah McIntyre and I, in order to come up with templates for what people can write in their methods section when they're doing this kind of small number of participants, and yet high statistical power, because we have so many trials and our effect sizes are so big. Provide templates to put into our method section, to put into our pre-registrations, so that we can really increase the credibility that the readers that we have in the mind of the reader, justifiably causing them to really believe that our results are not a false positive or a false negative. So I'm looking forward to hearing what others are going to say in this session about open science practices and pre-registration and so on. Thank you.